which acts like a drug. And obviously, in the beginning, they thought that gene therapy would be ideal for genetic diseases which are caused due to a mutation in a single gene. And the mutant gene can then be replaced with the wild type gene, which will correct the defect. Okay. So the first gene therapy trial in humans. Now, let me again go back a little and tell you what are trials. So before any new, um, just sorry. Yes. Okay. So before, just a minute, I think I will change this a little bit because you can't see me, right? Can you see me?
Ma'am, your video is
Yes,
off,
ma'am.
ma'am.
Yeah, I think I have to switch on my video and I have to go back to my. Um,
Yeah, you are now visible.
yeah. Okay. So I was talking about trials. So before any drug comes into the market, one has to do a lot of studies, which are called preclinical studies. And before you go to the clinics, you do studies, and then you have to do what is called as trials, clinical trials. So you have to start with phase 1 trials to show that whatever you are planning to use as a therapy is safe and it is not harmful to the patient. And then you go on to do phase 2, phase 3 trials in which you show that it's not only safe, but it is also efficacious. That means it is going to cure the disease. So now, if you look at gene therapy, it is still most of the gene therapy uh, techniques, they are all under clinical trials. So they have not yet, only a very few of them have reached approval from the Food and Drug Administration. So once you finish all the trials, then you get approval to use it as a drug and sell it across the counter in the market. So there are about more than 2,000 trials already which are going on for gene therapy. And these studies have provided the proof of principle to show that they are safe and effective. But very few of them have actually come to the market. But we'll see some examples which have come to the market. Now, if you look at the ongoing clinical trials on gene therapy, you find that most of them were for cancer. Unlike what was felt earlier, that gene therapy would be ideal for monogenic disorders. Monogenic means a single gene defect in the which causes a disease. So very few are for monogenic disorders, and most of them are for cancer. Because in cancer, so the problem is because in monogenic disorders you need the wild type gene which is introduced, the good gene which is introduced in the patient to remain forever till the life of that individual. Whereas in cancer, and that is one big problem so far, but whereas in cancer, you just want to kill the tumor cell and then the gene can be lost, it doesn't matter. So about 65% of the trials which are ongoing are for cancer and only about 10% are for monogenic disorders. So now what is required for gene therapy? Now, as I said, gene therapy requires a vector, a vehicle, which will carry the gene to the patient's cells, to the right cells. And also you need to know which gene to use. So you have to have the therapeutic or corrective gene, which you have selected and which you show that it is effective and it can cure the disease. Then next, you need stable and long-term gene expression. So you to show that the gene is being expressed at the cellular level as well as at the organ level. And also if you re-administer, so supposing a patient receives gene therapy once and then after a few months again he needs to get the injection, it should be safe to re-administer this gene and it should not be toxic. That means it should not be causing any harm to the patient. Now, there are different vectors which are used. As I said, vectors are like vehicles which carry the gene to the cells in the patient's body. And there are various different vectors which are being used. Adenovirus is one very commonly used vector. You'll hear a little bit more about it very soon. So these vectors, viral vectors, when they're used, they are made non-pathogenic by removing certain parts of the genome in the vector, which are harmful to the patient, which causes a disease. And in place of that, you put in the therapeutic gene. That means the gene that you want to introduce into the patient's body. So adenoviruses are one, adeno-associated viruses are another viruses. Then there are retroviruses, which are also used as vectors, and lentiviruses, which are the HIV viruses and liposomes, etc. So if you look at again the trials which are going on, most of the trials, or almost half of the viral trials, are using adenoviruses, and the others are using retroviruses. And also plasmid DNA can be used as a vector to carry the gene into the cells of the patient. Now in 1990, Michael Blaise, French Anderson, and his colleagues at National Institute of Health in USA, they carried out the first gene therapy trial in two children. These two little girls had a disease called as severe combined immunodeficiency syndrome, which means that their immunity was so poor that they were not able to live a normal life, because they would get infected very soon because their immune cells were not functioning well. And one of the reasons for this was that there was a mutation in the gene called adenosine deaminase, ADA gene. And this gene is absolutely essential for the T cells to function properly. So since there was a mutation in the ADA gene, the T cells were not functioning properly. And so these children were suffering from this disease called severe combined immunodeficiency. And then, so these patients were taken for the therapy in which the mutated ADA gene was replaced with a good wild type gene. And you can see these are the two girls who received this therapy about 12 years ago. So this is after 12 years, they're doing very well. And these girls would normally not have survived for so long because their immune system is very poor. But they could rectify this with the gene therapy, where they used a retroviral vector to introduce the wild type ADA gene into their T cells and then infuse them back into these patients. And they had almost normal immune system in their body. So these are the two, and these are the three men who first started gene therapy in 1990. They started the gene therapy thing, and they were Dr. French Anderson, Michael Blaise, and Kenneth Culver, who did this therapy. And this is a picture after 13 years. So you can imagine these kids who would normally not have lived at all for more than a few months were now able to survive, and they are leading a normal life. They're going to school like normal children, and they're leading a normal life thanks to gene therapy, where their ADA gene was replaced in the T cells, and the T cells became functional. But there was one problem with this, that these children, these two girls, had to go back to the hospital every 6 months to get an infusion of fresh T cells, because T cells, as you know, have a limited lifespan in the body. So they had to keep going back to the hospital to get fresh T cells, which were transduced or infected with the retrovirus carrying the wild type ATA gene. Now, so since they had to overcome this problem, what they thought was Alan Fisher, what he did was he used the stem cells, the hematopoietic stem cells, which are CD34 positive cells. And these cells he transduced with the retrovirus, which was carrying the ADA gene, so that these cells could be self-renew, and they would maintain the T cells which were having the wild type ADA gene. So this was quite successful initially, and they treated about 20 patients. But there was a problem. Out of these 20 patients who were absolutely cured of the disease, there were at least two who received. There were five who had some kind of a. They developed leukemia. That means the T cells proliferated much more than they should have proliferated, and in fact one of them even died, and so they had to really think about what is going wrong. Why is it that these uh, children are developing leukemia? And they found that it is because there is what is called as insertional mutagenesis. That means the retrovirus is getting incorporated in the host genome, in a place where it is activating a cell, a gene like the LMO2 gene, which is responsible for T cell proliferation, and the cells were just proliferating just like in cancer. So this was a problem, and of course later they did a lot of research on this, and they found out how they could stop this, and they could continue with this treatment of the CD34 positive cells with the retrovirus carrying the wild type gene. This is the picture of one of the young girls, Augustine, whose CD34 positive cells were transfected with using a retrovirus, and she's doing really well. And now she has normally functioning T cells. Of course, there have been problems also with gene therapy. So, as I said, adenoviruses are used to a large extent to transfer a gene. So, adenovirus in this case was used to transfer a gene called ornithine transcarbamylase in this young boy of 18 years old. But unfortunately, what happened was the dose of the adenovirus that was used was very high. And because of this, this young boy died because of lung and liver failure. So this was a sad thing. So again, they had to rethink about how they should overcome such problems of gene therapy, and they realized that one should first test the dose of adenovirus which is being given to the patient, and they should also see if there are any circulating adenoviruses in the patient's body, because adenovirus is a common cold causing virus which is there in most people. So one had to check whether this therapy could be given to such people or not. So again, as I said, that number of trials for cancer was very high compared to the others. So we'll see why cancer. I mean, cancer, you can use different strategies. You don't need to use just one gene. You can use different strategies to kill the tumor cells. So in most of the clinical trials that were going on, retroviral vectors were being used, and these retroviral vectors were based on mouse Moloney leukemia virus. So this is a mouse virus which was being used as a vector to transfer the genes into the human cells. And this was because the Moloney leukemia virus is very well characterized, its whole sequence is known, and it is known to work very effectively in animal models. And the gene system which was used with the retroviruses was for cancer was suicide gene therapy, which is using herpes simplex thymidine kinase gene. So what is suicide gene therapy? So let us just see what suicide gene therapy is. A suicide gene therapy is where there's a suicide gene. I'll tell you what this suicide gene is later. So this suicide gene is first incorporated into the viral vector. So here there is a vector, which is earlier they used retroviral vectors. And then this vector is introduced into the tumor cells. Okay. So once it goes into the tumor cell, it starts making an enzyme, and this enzyme is able to transform a prodrug, which is a non-harmful drug, into a harmful drug. So because of this enzyme, this prodrug becomes toxic and it can kill the cells. And along with this, it not only kills the cell which is expressing this enzyme, but it also kills the surrounding cells, because the active component which is toxic to the cells is passed on to the neighboring cells by a method called as bystander effect. And so there are more cells dying than the number of cells which are being transfected or infected with this virus. So what are these suicide genes? So these suicide genes are two types, two examples of suicide genes. One is the herpes simplex thymidine kinase gene, HSVTK, and the other is cytosine deaminase gene. How do they act? How do they kill these cells? So as I said, this is called pro-drug activation. So one of the suicide genes, one of the pro-drugs, which is not toxic to the patient's body, is ganciclovir. So ganciclovir is non-toxic, but in the presence of this enzyme HSVTK, which is the suicide gene, it will convert it into a phosphorylated form, GCVP, and this further gets phosphorylated, which becomes triphosphate, which gets incorporated into the DNA strand, and this is toxic to the cell because it blocks DNA synthesis, and that is how the cell dies. So this is one example of one of the suicide genes. The other suicide gene which is commonly used is cytosine deaminase. And cytosine deaminase is an enzyme from bacteria which converts a non-toxic 5-fluorocytosine. This is a drug which is not toxic. It converts it into a toxic form by acting on this molecule because of this cytosine deaminase, and this can kill the cells. So I'll just give an example of what we had done in our laboratory when we started work on gene therapy many years ago. So we wanted to carry out gene therapy for oral cancers. These come under head and neck cancers. So oral cancer is a cancer of the mouth. So for that, first we established a cell line from a patient who was suffering from oral cancer, and then these cells could grow as xenografts. Xeno means a human cell growing in a mouse. So these were grown in nude mouse, in a nude mouse model. So what are nude mice? I don't know if you've heard of nude mice. Nude mice are mice in which there are no T cells. So these are immunocompromised mice. And because they are immunocompromised, you can grow cells from any other species, like the human cells can grow very well in these nude mice. Then we also made this retroviral vector, which was carrying the suicide gene, HSV-TK, herpes simplex virus thymidine kinase. And then we characterize this, and we use this for our studies. So this is just a picture of the cell line that we developed from a patient who had oral cancer, and it's called NT80. And these cells can be grown in culture. They can be grown in nude mice, so they can grow as xenografts in nude mice. So we use this system to study our gene therapy preclinical studies. And this is just to cut a very long story short. We showed that when the tumor is injected with this HSV-TK gene, and then the mouse is treated with ganciclovir, then the tumor reduces in size compared to the mouse which is not treated with HSV-TK and ganciclovir. So this showed that HSV-TK and ganciclovir system could kill the tumor cells. And this is just the histopathology to show that there is a lot of cell death because of when the cells are treated, when the animal is treated with ganciclovir. So then we, again, I'm just going very quickly over this because this is not really, I mean, I just want to show you what are the kind of experiments that can be carried out in the laboratory to carry out all these preclinical studies. So we did a combination therapy of HSV-TK along with IL-2, and we showed that so this is, we always have to have a control, which is the untreated animal. This is a nude mouse. So these nude mice, they are not only immunocompromised, but they are hairless also. They have no hair compared to the other normal mice. So this tumor grows in the untreated mouse. Whereas if you treat them with just HSV-TK, there is no difference. But if you treat it with and with ganciclovir alone, the tumor still grows. You can see the tumor is really big in all three. But when you have HSV-TK as well as ganciclovir, then the tumor regresses a lot. But in the presence of IL-2, now interleukin-2 is a cytokine which not only stimulates the T cells, there are no T cells in these animals, but it stimulates the NK cell activity also. And so here you find that the tumor regresses completely. So we were planning to do phase 1, phase 2 trials with this system, but we realized that although retroviruses were being used in a large number of clinical trials, they had really failed to demonstrate this activity of cell kill. So this just showed that although your experiments can be really very successful in the animal models, they may not be just the same, the success rate may not be the same in human patients. So based on these trials, most of the people stopped working, most of the clinical trials on retroviral vectors were stopped, and they started working with another virus, which is the adenovirus. So we also started working with adenoviruses, and adenovirus is, as I said, a common cold virus, and we cloned the HSV-TK gene in this, and these viruses also carry the GFP gene, that is the green fluorescent protein gene. So we use this vector and we cloned our gene, and this is just to check So this green fluorescence protein is a dye which the virus carries. The gene for this is carried by the virus, and the cells then express this dye, which can be seen under a fluorescent microscope. And this is harmless to the cells, so the cells survive. But this just shows that the cells are infected or transfected with this retrovirus, which is carrying the GFP protein as well as the HSVTK gene. Now we did again animal experiments. So these are the mice which were before treatment. So you can see that the tumor has grown to a certain extent. After that, we gave them different treatments. This is the control in which no treatment was given. And these are the two mice these are mice which were injected with different doses of this adenovirus vector carrying the HSV-TK followed by ganciclovir. And you can see that the tumor has regressed completely. It did not grow like in the control. So this again, we were very excited about this, and we thought we should do a clinical trial. But again, we had seen from the ongoing clinical trials that there is very limited transduction of the viral vectors, adenoviral vectors, and there's low expression of thymidine kinase gene. So we wanted to increase this uh, uh, the expression as well as the infectivity. And for that, again, I'll just cut a long story short. We used a histone deacetylase inhibitor, HDAC inhibitor. And we found that when we use this HDAC inhibitor, like valproic acid is one of the HDAC inhibitors, there is not only an increase in the number of cells which are transfected with the adenovirus, but also the expression of the adenovirus, of the green fluorescent protein which the adenovirus carries goes up. So we thought that this would be a good system in our clinical trial. Again, we showed that the tumor regresses in these cells. So based on that, we wrote a phase 1, that is very early stage clinical trial protocol for using it in the patients, using this adenovirus carrying the HSV-TK gene, followed by ganciclovir, along with valproic acid, which is the HDAC inhibitor. But unfortunately, there are many regulatory issues which come up when you're doing a clinical trial, and somehow this was not really possible to carry out a gene therapy trial. But we did all the preclinical studies that were required to show that it is safe and it is effective. Now we'll move on to the gene therapy world scenario. What is happening in the world? So the first drug, which, as I told you, that you have to do phase 1, phase 2, phase 3, and only after that the Food and Drug Administration gives permission. If they are convinced that it is a safe drug and it is effective, then only they give you permission to use it in patients on a charge basis. That means you can buy it as a drug. So the first drug which got permission to be used uh, as a drug for gene therapy was called gendicin, and this is an adenoviral based P53 gene. P53 is a tumor suppressor gene which is mutated or lost or modified in a large number of tumors, because of which the tumor keeps on proliferating. So in China, they use this. They got permission to use this as a drug for head and neck cancer cells. So gendicin has also been used along with as an adjuvant therapy, along with chemotherapy or with radiation therapy. And another drug which they developed in China and for which they got the permission was oncorin, which was also used for squamous cell carcinoma, that is for oral cancers. Then these are other examples of gene therapy drugs which have come into the market. As I said, there are just very few, about 5 or 6 of the drugs which have got permission to be used as a drug. And this is one which is Glybera, which is the name is, it's also called alipogene tiparbobic. And this is again adeno-associated viral gene delivery, which codes for an enzyme called LPL, lipoprotein lipase. And so there are patients who are deficient in lipoprotein lipase, because of which they have elevated levels of serum triglycerides causing life-threatening pancreatitis. So for such patients, using this gene therapy product, they could bring down the level of lipoprotein, serum triglycerides, and reduce the chances of getting pancreatitis. Then another drug which was used, which was given permission by the US FDA as well as in Europe, was Imligic, which was used for uh, this is the first approval of a oncolytic virotherapy. That means the virus enters into the cell and it can divide, but it is not toxic to the other cells. It is not toxic to the cells. And this is based on the vector carrying GM-CSF gene. So this was the first drug which was used for as immune response against infected tumor cells. Then another drug which was approved, as I said, autologous CD34. These are the hematopoietic stem cells which could be transfected with the virus carrying the wild type adenosine deaminase gene, ADA gene, and this was approved for treating patients who had immunodeficiency, the severe combined immunodeficiency disease. This is just a company which is making that the product. And then there are many trials which are still going on, which which look very promising. Um, Again, I don't think I have time to go into all the details of these non-lytic replication component retroviral vectors, but just very briefly, I can tell you that there is a drug which is, uh, they have called it TOCA 511, which is injected into the brain for brain tumors. And this drug this virus is such that it can replicate only in the tumor cells and not in the normal cells. So if you inject it into the tumor, it will proliferate in the tumor, but not in the normal cells. And this virus carries the suicide gene, cytosine deaminase, which I told you earlier, which converts 5-fluorocytosine into a toxic form, which is 5-fluorouracil. And that is how it can then clear all the tumor cells. So this is one study which is going on. So what they found in a clinical trial is this helps um, to, the patients survive. Normally they would have survived for about 7 to 9 months. But here, after this treatment, the patients survive for at least 24 months. So that is what they think is really helping the patients with brain tumors. Now, there's another uh, gene therapy product which is very much spoken about. Even in India, a lot of work is going on this. And this is personalized cell and gene therapy, which is development of CAR T cells. It's called CAR T cell therapy, which is CAR T stands for chimeric. Chimeric means two different things coming together. You call it a chimera. So chimeric antigen receptor T cells, and this was designated as a breakthrough therapy by US FDA in 2014. And this therapy is used mainly in acute lymphoblastic leukemia patients, where the T cells are removed and then they are genetically manipulated. That means they are changed, they are transfected with genes which will express certain proteins. And then these cells are grown in large numbers, which are genetically manipulated. And then they are transfused back into the patient so that they will target the cells which are expressing CD19 protein, which is present on the tumor cells. So it will get rid of all the tumor cells. And they found that in the clinical trials, they found that 89% of the ALL patients, acute lymphoblastic leukemia patients, went into remission. That means they no more showed the presence of these tumor cells in their body for more than 2 years. So again, as I just said, these T cells are collected from the patient's blood, and then they are genetically engineered to produce special receptors on their surface called chimeric antigen receptors, CARs. Okay, chimeric antigen receptors are expressed on their cell surface, and these chimeric antigen receptors, they recognize certain antigens on the tumor cells, and then so this is done outside the body. This is called ex vivo therapy, where the cells are taken from the patient in the culture medium. They're cultured outside. They're transfected with cells which are carrying these CARs, or the chimeric antigen receptors. And then these engineered CAR T cells are then grown in the laboratory in the presence of interleukin 2. They are expanded and then they are infused back into the patient. And after the infusion, the T cells multiply in the patient's body and they recognize and kill the cancer cells that harbor the antigen against which these scars are made. So this was a young girl who was the first patient to be treated and cured of leukemia with CAR T cell therapy way back in 2015. So finally in 17, so as if you remember what I said, that clinical trials are carried out, and then finally the Food and Drug Administration has to give approval so that uh, so that it can be used as a drug or as a therapy in the clinics. So this is very important to get this Food and Drug Administration approval. So this was the first gene therapy to be approved by the United States Food and Drug Administration. So it's called CAR T cell therapy. Now I'll talk a little bit about. We all know today there's a lot of talk about different vaccines which are being used for COVID-19, and I'm sure all of you have heard about Covishield and Covaxin, and all these different vaccines which are being administered to um, healthy individuals in our country. So let us just see. That is also a part, it is a form of gene therapy that is being carried out. So COVID mRNA vaccines, you must have heard about mRNA vaccine. M stands for the messenger RNA. So mRNA vaccines. So let's see, what are these mRNA? So the COVID mRNA vaccines, they carry a mRNA, that is a messenger RNA, which codes for one of the spike proteins. You know that the virus has spikes on the outside surface, and when a person is infected with this SARS-CoV-2 virus, then they develop antibodies to these spike proteins which are present on the cell surface of the virus. So now we can make these vaccine carries mRNA, which makes these spike proteins, and then and how is it given? This mRNA is given in oily bubbles coatings made of lipid nanoparticles, which can fuse with the cell surface and enter into the cell. So once it enters into the cell, the mRNA will start making the spike protein, which can fight the virus. So the immune system recognizes the spike protein as a foreign invader and produces antibodies against it. So if the antibodies later encounter the actual coronavirus, they are ready to recognize and destroy it before it causes the illness. So this is how the mRNA vaccine works. So mRNA makes the spike protein against which antibodies are made in the body, and these antibodies are then ready to fight against any coronavirus which enters into the body. So these are two vaccines, Pfizer and Moderna, which is making these mRNA vaccines. And I said, as they are against the spike protein of the SARS-CoV-2 virus. And these have been granted emergency use authorization by US FDA, and they are being administered to all the individuals in the USA. In India, we are getting what is known as Covishield, which is a vaccine which is made by Serum Institute of India in Pune. So this is based on the Oxford vaccine, which uses adenovirus. But this adenovirus is from the chimpanzee. So that's why it's called chimpanzee adenovirus. And as I told you earlier, adenoviruses are the viruses which are responsible for common cold, and they are not harmful to the individual at all. So this adenovirus has been genetically modified so that, first of all, it is safe. It does not cause any disease, and it is impossible for it to grow in the humans. And this carries the gene which will code for the spike protein. So once this virus is injected into an individual's body, it will start making the spike protein, and again it will work in the same way. There will be antibodies raised against the spike protein in the body, and this is what will give immunity to the individual against the coronavirus. Okay, now we'll move on to two techniques which have been a boost to cell and gene-based therapies. One is inducible pluripotent stem cells. Yamanaka got the Nobel Prize for that. And the other is gene editing, for which two ladies have got the Nobel Prize recently. I don't have time to talk about indicible pluripotent stem cells because that itself is a big topic, but I'll talk a little bit about gene editing techniques, including the CRISPR-Cas9 technique. So first of all, what is genome editing? So genome editing, uh, it's a group of technologies that give scientists the ability to change an organism's DNA. So you can either remove, add, or alter the DNA using this technology. So let us see how that can be done. So earlier genome editing, which actually brings about double strand breaks, so DSBs, double strand breaks. So the molecules which were used earlier were meganuclease. Just one second, please. Sorry. So the double strand break inducing nucleases such as meganucleases, zinc fingers nucleases, transcription activator like effector nucleases called talins. These were used, which target specific sites in the genome following protein-based systems. So they could target to specific sites and cut the DNA. So, but recently now they're using what is called as a CRISPR-Cas9 associated system. So CRISPR stands for clustered, regularly interspaced short palindromic repeat sequences. And for this, you require a RNA-guided nuclease. And this has really revolutionized the way the genome editing is performed. So these were the different examples. I said meganucleases, zinc finger nucleases, talins, and CRISPR-Cas9. So these three are the ones in which nucleases are used to specifically cut DNA at known sites. And CRISPR-Cas9 is another method in which, again, you can cut DNA at very specific sites, and then you can introduce a target DNA into this place where homologous recombination takes place and the two ends are joined again. So it can carry a mutation or an insertion or introduce a piece of DNA in this broken site. And so these are the two ladies who got the Nobel Prize in 2020 in chemistry. Uh, so this is Emmanuel Charpentier and Jennifer Doudna, who developed a method for gene editing. This is using the CRISPR-Cas9 system. So CRISPR-Cas9 was adapted from a naturally occurring genome editing system in the bacteria. So when a bacterium gets infected with a virus, then what it does is it cuts off small bits of the DNA from the virus and forms a CRISPR array. And this array is a DNA piece of DNA which allows the bacteria to remember the virus. So if the virus attacks again, then the bacteria produces RNA segments. So the DNA is translated into RNA segments from the CRISPR arrays to target the virus DNA. And then the bacteria uses Cas9, which is an enzyme or a similar enzyme, to cut the DNA apart, which disables the virus. So this is what is present in the bacteria, and this was used for gene editing in the eukaryotic cells. So what happens in the eukaryotic cells, what is done is a small piece of RNA with a short guide sequence is created. And this guide RNA recognizes very specific target. So it depends on which region of the DNA you want to cut. Based on that, you make the guide RNA, which carries very specific target sequences for the DNA in the genome. And then the RNA also binds to the Cas9 enzyme, which is introduced into these cells. And the modified RNA is used to recognize the DNA sequence, and the Cas9 enzyme cuts the DNA at the targeted location. So once the DNA is cut, then the researchers can use the cell's own DNA repair machinery to add or delete pieces of genetic material or to make changes to the DNA by replacing an existing segment with a customized DNA sequence. So that is how you can change the genome by using this CRISPR-Cas9 system. So this just shows, again, this has been taken from the net. This shows that when a phage DNA enters into the bacterium, then the invader DNA is cut into pieces, and this goes and sits on the CRISPR locus, and then this is transcribed into a pro-CRISPR RNA, and then this is processed to give you the CRISPR RNAs, which are made in the bacterium and kept there. And then once they encounter the same DNA, the same phage again, then it goes and binds over there to specific sites and cuts the DNA, and that's how it takes care. So this is a kind of defense mechanism for the bacterium. So in Cas9, CRISPR-Cas9, CRISPR is associated with the Cas9 protein, and the RNA guided DNA is made along with the CRISPR-Cas9. Um, it forms this molecule which can then recognize very specific sites on the target DNA and cut it with the Cas9 system. And this is now being used for genome editing. So as I said, this Cas9, it searches for the targeted DNA along with the guide RNA, which is incorporated in that. And then it recognizes the site which it wants to target, which we can identify. We can cut it at specific sites. And then this is how genome editing is carried out inside the cell. Again, this just shows how the guiding piece of genetic material along with the Cas9, it enters into the cell and it specifically cuts the DNA at sites where we want it to cut. And then they are finally rejoined by homologous recombination inside the cell. So this is how it cuts, and there's repair mechanism. And then you can either introduce a piece of DNA into the thing, or you can just correct it, or you can bring in a mutation and you can do whatever you like. And this system is really very simple to use. So here is an example where they made a chimeric mouse gene targeted for the agouti coat color gene. So this was a normal brown colored mouse. And in this, they have made a chimeric mouse which targeted the coat color gene and made it into a agouti color. So this light brown is what the agouti color. So this shows that gene editing can be used in vivo as well. So there's an interesting story about gene editing based therapy for AIDS. Now there was this person called Timothy Brown, and the story is now known as the Berlin patient story. So what happened was Brown was suffering from acute myeloid leukemia, and he also had HIV infection. And because of this AML, he received a bone marrow transplant from a donor. And at that time, they didn't know that this donor had a homozygous deletion for a naturally occurring 32 base pair deletion in the CCR5 gene, which is a co-receptor for HIV-1. So because there was this homozygous, because there was a deletion in this gene, so it could not act as a receptor for HIV-1. Now, following the transplant, so this patient, Brown, received this blood in which the donor blood, the cells that he got from the donor individual had this deletion. And following the success of transplant, Brown continued HIV therapy and had no detectable viremia for over 6 years. So this is because he had received blood, he had received cells from the patient, from a normal individual who had this deletion, and it was not known at that time. But because of this deletion now, with this bone marrow transplant, his blood had all these cells which had a CCR5 deletion, because of which HIV could not infect these cells. And so he was sort of cured of HIV. So this observation has led to CCR5 modification, which has become a gene therapy target for curative HIV research, where by gene editing, you can bring about this deletion in the CCR5 gene for homozygous deletion, and the cells will no longer be able to be infected with HIV. So by correcting disease-causing mutations, it may be possible to reverse disease symptoms. However, in order to optimize the therapeutic potential, the appropriate delivery specificity and repair strategies must be established. So there are still quite a lot of problems with gene editing because it can be off-target editing, which is not what one would require. So one has to be very careful about that. Um, there are papers where people have used CRISPR-Cas9 mediated genome editing of epigenetic factors for cancer therapy. So, in tumor related epigenetic alterations or epigenetic factor mutations play a major role during the various steps of cancer formation or carcinogenesis and affect a variety of cancer related genes and a wide range of cancerous phenotypes. So therefore, epigenetic regulatory enzymes may be good candidates for cancer therapies, what they have reported in this paper. So with study improvement in functionality, delivery, and specificity, it is now possible that CRISPR-Cas9 system will enable the development of necessary treatment options for genetic diseases and affect a variety of organs and tissues. So there have been very rapid advances made in this CRISPR-Cas9 technology, as well as in the delivery modalities for gene therapy applications. And this is now becoming more and more reality in treatment. So there's this scientist Feng Shang who worked a lot on CRISPR-Cas9, and he says that this exciting development of CRISPR-Cas9 technology for genome editing, this also raises a lot of societal challenges. So one has to be really careful and cannot misuse this very powerful technology, uh, and hope to use it responsibly and carefully. So finally, the future of gene therapy and gene editing, these are emerging as promising ways of treating diseases. And the successes continue to be achieved in experimental models. As I showed you, adenoviruses are now being used in the vaccines. The COVID vaccines are using adenoviral vectors. Besides that, adeno-associated viruses and antiviral vectors are also showing great promise. And there are a large number of pharma industries now which are making strategic investments in manufacturing the gene therapy products and carrying out clinical trials. So we have learned a lot from many of these clinical trials, what we would never have learned from animal studies alone. Something that works in animals doesn't necessarily work in the humans, is what we learned. And now it is time to harness the power of gene therapy to treat cancer and some of the rare diseases. Thank you. So I will now be ready to take any questions.
Thank you so much, ma'am. So, participants may please raise your questions by unmuting yourself.
Are there any questions? I think there are very few people who are attending.
Yes, ma'am.
Yesterday, there were a lot more people attending.
Yes, ma'am. So, if you have queries, dear participants, please jot out it in the chat boxes. And thank you so much, ma'am, for a very informative session on personalized medicine for cancer. And you have explained suicidal gene therapy by quoting few drugs and their pharmacodynamics as well, ma'am. And you have rightly pointed out the present scenario on COVID mRNA vaccines. And even you have demonstrated few pictures of the people who have underwent the gene therapy. And you explained very clear concepts on gene engineering therapy for AIDS treatment and CRISPR-Cas9 technology and future of gene therapy. Thank you so much, ma'am. It was very informative, and it is very pleasure for us to have you with us, ma'am. Thank you so much.
Ladies and gentlemen, let us dive into the world of fungi now. I kindly request Honorable Dr. Ahmed Abdul Halim Khan sir to take over the session.
Thank you. Thank you, Rita Madam, for such a wonderful, talk on gene therapy. Always you rejuvenate the talk, and this time you have also included some part of COVID-19 also. So it is a great time. We again had online virtually. So again, once again, a great big thank you for accepting the request. Whenever we request, please, ma'am, do help us in such virtual events also. As we know the restriction of time that we couldn't have the offline mode workshops or your meetings. So like this also, please bless us and make us to motivate many more women people. So your presence, your presence and Susan madam's presence make us that really women are still strong, and they are the important pillar of the society, and this society is also empowering the women. So like this, we feel very proud, and we are very wholeheartedly thanking Susan ma'am and you for accepting our invitation. Though we equally thank Surendra sir also for his support. Without his convenership, it was not possible for us to make this event so wonderful. So thank you, ma'am. Thank you so much for your such a sound presentation. Thank you so much, ma'am, again.
It is my pleasure, Dr. Khan. Whenever you request us, we cannot say no, because we know it's for such a good cause that you're working so hard. So all the credit goes to you.
Thank you so much, ma'am. Thank you. It is all you people are making us to grow slowly. In a slow speed, we are growing by looking you. So we need more and more, blessings and empowerment from your side in all ways. Now it is a time for my turn, and I think I will be making today's presentation a little bit more beautiful and more attractive. In what way I will be making? Please let me share something. What are fungi? Fungi are usually the commonmost organisms studied by botany people, and equally it is also studied by the. It is also being studied by all the life sciences, almost all the life sciences, in medical mycology or in various forms it is studied. So fungi or the organisms usually having eukaryotic organization or well developed, earliest well developed form of eukaryotes. And the special thing is they have the chitin in their cell wall, and this um, fungi these fungi are a diverse microbial community separated from the bacteria, archaea, and protista as a separate kingdom. And Blackwell in 2011 has discussed the classification, discovery, identification of fungi, and various estimates have been done in various times. You can look at the table, and you can see these are the different times. In different times they have enumerated this kingdom with a different number of taxa, like Hawksworth in 1991, Hawksworth in 2001, and Brian in 2005, 6, and 2011 Mora, and Blackwell again the Hawksworth in last 2017. We can see that there are various numbers have been given here, and ultimately it is told that 5.1 million taxa are there among the fungi. Why I am going to tell about fungi is because these fungi are having the role of both the positive and the negative role. So here comes, we can see the green shaded circles are there and red shaded circles are there. So green shaded circles are plenty, which are showing us the beneficial role of fungi to mankind, and red is the one which is showing us the harmful or negative impact on the society. So as these fungi are known to produce so many things like antibiotics, alkaloids, biocontrol agents, enzymes, organic compounds, and plant growth regulators and pigments. But equally they are well known for having the different kind of diseases like now I am going to list out some of the diseases and I will be showing you what was the impact. And fungi are known to be the important, let me say, contributors in the industry of enzyme, which is a billion dollar industry. And India also contribute, or India also use the fungi and make number of enzymes in our country, like amylases. Amylases are the one which are made for a large scale on fermentative system, and other enzymes are also listed over here. A number of enzymes are made by using fungi as the source of the enzyme. And now we can see the most popular form of fungi, or you can say that whatever the name, study of fungi is called usually mycology, and the name mycology comes from a word called myx. The myx word is usually to signify the macroscopic form of fungi called mushroom. You can see here the mushroom, which is shown as the button type of things or fruiting bodies, which are found on the dead decomposing kind of organic matter. And here we can see the lot of mycelia is going to be under the surface of the substratum. And like this, these are the beautiful macroscopic form of fungi which are used as the food among the mankind. So this is the fungi which is going to grow on the moist wood, and it is going to give a typical beauty to the wood, decomposing wood which is in the water. And you can see, the as I just now told, that they are also important role in this foliages. So here we can see the number of fruits and the rotting part, whatever the rotting part is there, as saprophytes. They'll be growing on different types of particularly food items, and they will be growing in this kind of typical velvety appearance on the surface with the fruiting structures. Again, these we can see the penicillia, which is having conidia and conidiophores, then phyllites and the branch type of verticillate, monoverticillate, and all these kind of things will be there in the details of the structure. And then we can see just now we have seen the largest type, or the largest microscopic, which is easily visible by naked eye. And we can see the smallest forms which appears in a huge colony and rot, we can see with the naked eye. And we can see these are the smallest form or unicellular forms. So here we are making fungi into two types, like we can say them as to be the particularly molds, which are to say filamentous forms, and unicellular forms are also there, like yeast, which are the single cell organisms. And well known that from Egyptian civilization, or more than 5,000 years back, the baker's yeast was used in making breads, and also as well as the beverages made for the alcoholic type of drinks. So here my title is friends or foe. I'll be talking all this, what is this deadly, what is friendly, what is delightful. So in this case, first we'll begin with the friendliness or the life saving forms of fungi. Like we can see penicillium is the most important fungi what has been discovered by the Alexander Fleming. Here you can see the picture of Alexander Fleming holding a plate which is used for the cultivation of Staphylococcus aureus. So when you see the Staphylococcus aureus is a gram-positive bacteria growing in the particularly spherical in shape and grape bunch-like appearance. And suddenly, when you see the plate holding in the Fleming's hand on the other side of the pose, we can see that there is a huge cottony mass growing on one end of the plate. So the thing is, the bacteria and the fungi is a combination in the plate itself. In the same single plate, we can see there is a white cottony mass growing on the surface of the plate, and there is one more creamy type of the growth which is growing in a zigzag pattern as it is streaked on the plate. So here we can see that there was a halo around the particularly cottony mass. The halo around the cottony mass is expected to be the zone of inhibition, that the fungi growing on the one edge of the plate was not allowing the bacteria to grow. So this was the they say serendipitous discovery of penicillin or antibiotic for the first time. So in the field of chemotherapy, this was the biggest they say achievement that was obtained. Though chemotherapy was having the drugs by salvarsan and protonsil like drugs were there, but still the drugs were made from arsenicals and mercurials. Now the drugs of this variety were the first time drugs made, and they are free from metal salts. Earlier drugs were metal salts and having lot of side effects when utilized, but penicillin or penicilliums, whatever the metabolite is there, the metabolite was named antibiotic, and this was the first time. Why this antibiotic has got so popularity is the biggest question mark. So now I'm going to discuss about that, why it is got the popularity. You can see here the again the I'm going to say hand-drawn picture of the I'm going to say penicillin, where we can see the plate yellow portion is nothing but expected to be the bacteria, that is Staphylococcus aureus, which is well known to be growing in the great bunch-like appearances, and a zone is having a clear halo surrounding the penicillium notatum, and this is the a hand-drawn picture of Alexander Fleming in his laboratory. Okay, and this is also one girl. I mean to say it is a just I mean to say hand-drawn picture. I mean to say amazed. Just a girl is looking very amazed on the penicillin discovery. How it has suddenly come in the, that is the sudden the shocking face on the girl's face that is shown on the picture. Why this penicillium? Okay, we can see the picture of penicillium, how this penicillium looks like. The penicillium looks like this on the agar plate. Whatever the agar plate was held in the hands of Alexander Fleming is shown over here. How the pigmentation, like you can see the gray pigmentation, and you can see the electron microscopic pictures of the penicillium, which is having a paint brush-like appearance. Paint brush-like appearance is nothing but this is the particularly conidia, and these are the conidiophores and sterigmata, and the paint brush-like appearance have made this to appear. I mean to say the name has come penicillium. Why this penicillium has got such a big news? Because the penicillin was the miracle drug at the time, because World War II was going on. At that moment, when Alexander Fleming has discovered this penicillin, the scenario of the world was World War II was going on, and in World War I, we can see that a lot of soldiers in the war have died just because of their wounds were not properly healed. Because of the sepsis, or we can say septicemia, the patient, I mean to say, a lot of soldiers lost their life, and big loss was there to the mankind at that moment. But during the, I mean to say, World War II, the Alexander Fleming's discovery was a miracle drug, and it helped the soldiers like anything. And 95% of the soldiers who were severely wounded after administering the penicillin, you can see the picture. Again, a handmade drawing where it is shown that a soldier is being administered with the penicillin. So 95% of the soldiers at that moment were healed, and they could become fit from their wounds. And compared to First World War, the First World War had big loss of soldiers, but here only the 25% of the people were died or lost their life because of some kind of medical ailments. So that is how this made the penicillin miracle drug, and penicillium as a hero, and Alexander Fleming as a Nobel Prize winner with other some his colleagues. And now, do you believe that biggest challenge what we have seen just now is a fungus has now saved the life of many people. That is the good thing what we have seen now. Now the thing comes is, do you believe that fungus made people to immigrate? Immigrate means what? To move from one place to the another place. So this was also has happened. And now I am going to show you one of the picture. Do you know who he is? He is none other than the present president of America. Why I am telling suddenly about fungi and American president? The thing is, because he is the American president Joe Biden, and not only Joe Biden, but there are 23 presidents of United States of America who are from Ireland. Why I'm talking all this thing about the Ireland? So please remember Joe Biden and other people. And just before Joe Biden, we had one more president called Barack Obama. So Barack Obama's ancestry is also from the particularly our Ireland. So more than 23 presidents, of United States of America were from Ireland. So what happened in Ireland? Why these people like Joe Biden or their ancestors, before president ancestors, are found to come to America? Why they have Ireland is somewhere in Europe, and America, United States of America is in North America, and between we have a big ocean. And why they have traveled? Why they left the why they left the Ireland, and why they move? Why they move from Europe? Means I'm going to say Europe is very far from America. So how could they move from such a long distance, and why they have made why they were made to move from this place to another place? So that is a beautiful concept, or we can say that is a beautiful fact what we have to see. In the 1840s, we can see that there was a great famine. What famine? Famine is nothing but it is the scarcity of food. A famine is a kind of here in this consequence, famine is a condition where we can see that there is a big, I'm going to say, fight. There is a big fight for the food. Food was not available, though money was there. Like just sometimes back, we heard that in France or Italy, people were throwing their local currency in the streets by saying that this currency is not able to save their life. Similarly, a famine was there where they didn't get food. What was their food actually? Their food was potatoes. Like we eat rice or wheat over here, or maize, or whatever locally available cereal is there, we use that. But in Ireland at that time in 1840s, potato was the food. I mean to say, instead of cereal grains, potato was the food. So what happened to potatoes? So the people in Ireland couldn't get the particularly potato. Why they didn't could get the potato? That and all we'll talk. But here in these two hand-drawn pictures, we can see very clearly. One is a person who is going on the horse with his luggage, and one is a family where we can see that people are looking towards the potatoes or cooking kind of whatever thing is there. It is a still where we can see in the still there is a particularly what to say potato cooking is going on, that is shown here. So what happened to this? So people used to grow this potato. You might be knowing, not knowing, I don't know, but potato is a crop which is from Mexican origin. In Mexico, potatoes were actually origin. The crop of the potato plants are basically origin from the particularly Mexico. And here this Mexican crop was slowly fled to the various parts of the world. And in Ireland, there were two high yielding varieties of potatoes were there, and they were having a big shortage of potatoes because of a disease. What was the disease? I will tell you. But before that, potatoes were not at all available. As I told you in the initial part, that whatever the potatoes are there, potatoes were grown and potatoes were used as a food. And consecutively during 1845 to 1850, there was a big problem. There was no crop, or whatever the yields are there, the crop yields were zero. And you can see the women in a picture is shown digging the soil for getting some kind of food, and the baby is also shown in the picture where the baby's mother is digging the soil and the baby is collecting the food in the form of potato. So what has happened due to crop failure? There was a lot of problem. What was a lot of problem? People started, I mean to say, there is a scarcity of food, and in this pandemic also we have seen that a lot of people started begging. In the current COVID-19 time also, we have seen that there was a scarcity of food, or there is a scarcity of money, or same pandemic, whatever pandemic was there. Like in a pandemic, what we have seen, same situation was found in Ireland. People were, I mean to say, the people were started begging for the food, or people started riots, means they were doing decades in the people who have food, we don't have food, that kind of, you can see the picture where we can see the people are in a row for getting the food. And Here is a picture where we can see that the people are fighting for the food, and here also we can see they are they are burning the in anguish, or we can say in anger, they are burning the shelters of neighboring families for the food. So this was a big total overall. This potato non-availability of potato or not availability of food in Ireland has created such a big kind of issue, and they have told that over a 15 years of period, 1 million people died due to starvation and a disease, and people who died in a country, they died, and people who don't want to die, they slowly started moving from this place. And like this, they have told that 1.5 million people moved from Ireland. This is a story that immigration has occurred. People moved from Ireland to the various countries. Like that, a huge population of Ireland has flew, or we can say fled to the various means of transport. They go to the various parts of the world, and one such place is the United States of America, where we can see today the Irish, I'm going to say Irish origin or ancestry. With Irish ancestry, we have lot of presidents. You can guess, when they're presidents, you can also guess what amount of population could be accounted in United States of America, which fled from Ireland during that famine time. So all these sources are from a Encyclopedia of Britannica published in 2002. The title of this in the encyclopedia is Plant Diseases, Their Biology and Social Impact. You can even check out this issue and you can get the information. And here in this picture, one of the mother is, I'm going to say, praying or begging for the sake of food by holding a small child in hand. And people have packed their luggage and they are planning to move like that. That is a discussion given over here. What was the reason for this all story? The entire story of immigration and immigrants is just because of the famine. Potato scarcity was just because of potato plants were affected, or potato crop was affected heavily by the fungus that is called Phytophthora infestans. In the both pictures, you can see in this still you can see the visibility of your plant affected with the leaf with the symptoms of Phytophthora infection, and this disease was named. This disease is named as blight of potato, late blight of potato, and the causative agent is here shown, that is the Phytophthora. And both the Phytophthora destroys the leaf part and the tuber part of the potato plant. And one more problem is there, that is called holy fire. What is this holy fire? We'll talk. In the holy fire, we can see there is again one more book called The Salem Witch Trials, an unsolved mystery from history. It is a book, you can see this book is a beautiful presentation of the problem again caused by fungi. So here you can see there is a victim. You can see the name of the title is Witch. Witch means nothing but a lady who performs magic or black magic. A black magic performing lady is called witch. So here is shown the witch, and here shown is the particularly the solicitor, and we can see he's a judge, and here is the baby girl who is actually having the symptoms of the some typical symptoms. I'll discuss the typical symptoms. So this is a beautiful book written by these people. You can see here the people who wrote this book, like Jane is there, Heidi is there, and Yolan is there. You can read this book and you can come to know details how the problem was happened. So this is the picture again, a court trial room where the girl is showing typical, I would say, the girl is falling on the ground and having typical kind of, I would say, convulsions or typical behavior, strange behavior of the girl was depicted over here in this picture. You might be knowing that people who have black magic or effect of black magic, how they fall on the ground and how they do nuisance or what kind of, whatever the symptomology you can, you know very well that in still villages of our hamlets or villages, we can see same things are going to happen. So here, this is the lady who is actually the culprit, made as a culprit, and this is the advocate, and this is the judge, and this is a courtroom where it is shown that in Salem the girl of in between 9 to 17 years were having this, I mean to say, problem. How these big people have got a problem is a big question mark. These people have got the I mean to say problem just because of eating the food which is actually poisoned food. So at that moment, people didn't had any kind of guessings that what happened, why this kind of typical I mean to say magic type of whatever the problem was there. People not without the science was not so progressive at that moment, and the people did not guess what could happen all this. So like this it has happened, and then uh, this all I mean to say was a new kind of problem in that time. And they told that this place that is Salem, it is a British colony where Massachusetts witchcraft hysteria, I mean to say, just because of black magic people are having hysteric, and it resulted in jailing of more than 150 people, okay, and two dogs were died. And this trials happened not only in America, but also the same situation was found in the Europe. And people told that more than between 1 to 9 million people of Europeans were executed for witchcraft, and 15 this all were happened during the 1500 and the 1600s. So this is a picture where the innocent lady was probed, or she was made jailed. She was made sentenced just because of it is suspicion that she is a witch. She's making some black magic on the young girls. So what was this situation called at that moment? This situation was called Holy Fair in some time like 994, and Saint Anthony's Fire in 1093, and Salem witch trials in 1692, and in between the 1777 it was named as Sologne, and 1943 it was 1951 till 1951 this was a common kind of problem. And you can see that the name in this problem was called fire. Why fire? This is a painting made by Matthias showing that what happens actually when the person consumes poison containing grain. So this is the what skin gangrenes. The person will have gangrenes on the body initial stage, and then the gangrene will have a fire-like burning sensations in the body. What was actually happening to all these people? What was actually happening to all these people was just because of the grain what they used. Earlier I told you that people used to have the potatoes as their food. Now here again one more concept is people used to have the rye, rye, rye. Okay, so here it is the grain which is almost similar to your wheat type but not exactly wheat. So these grains were actually attacked by claviceps, and the claviceps was known to produce the what to say typical kind of toxins. And when these grains are to say made in a flour, and this flour is consumed in a bread form, then the toxin will have the typical to say the symptoms. Here you can see the typical symptoms of the people which were covered in the newspaper of that times. And here you can see the person examining the I'm going to say poisoned food. And this is also one of the picture where pond saint spirit it was there in 13th century. Likewise here the illness is covered in the newspaper at the time. And now you can see again an old man in having typical madness kind of symptoms because of that kind of poisoned food consumption. So what was the reason for this? The reason for this today we can call it as ergotism, and that was the reason. Ergotism, conversive ergotism is a reason, and ergotism was a reason for all this typical kind of I would say symptoms in the people. So ergotism is generally because of the poisoned kind of grains, as I told you. So here ergotism is generally nothing but it is containing some typical alkaloid in it. The grain is having the alkaloid, and the alkaloid is made by the pathogen that is claviceps. And if the alkaloid content is more than 2% or up to 2%, then automatically the person will start behaving typically like a mad kind of person. So here the symptoms could be divided into convulsive symptoms and gangrenous symptoms. Gangrenous symptoms lead to have the burning sensation in the body, and convulsive symptoms generally make the person to behave mad. And this madness was expected to be just because of the typical, I'm going to say, the madness was considered as to be the particularly magic type of the thing is there. The convulsions were considered as to be the particularly madness among the people. So people used to have the hallucinations. You know, knowing very well the what is hallucination. So this has hallucination is generally just because of the lysergic acid. Lysergic acid. It is a typical kind of compound which makes the person to behave in an ill form. And they also have the particularly the muscular contraction in them. Okay, so seizures are abnormal. Seizures are the symptoms of abnormal electrical activity in the brain. That is the why the people, the young girls, were affected by this lysergic acid or LSD, and this were named as clivin alkaloids, and which were causing the typical convulsions or seizures, and that is how the people were having the problem. So these are all the symptoms, and this is again the historical thing. And now you might be very knowing that the same whatever the I'm to say toxicated grains were once upon a time causing such a big problem. Today they are when you say after that they were used for inducing the easy labor. Labor means I think most of them are ladies. They might be knowing. I mean to say delivery pains, whatever the pains in pregnant women for termination of pregnancy, whatever the pains are coming, they are called labor pains. So, if 2 or 3 grains are given to the pregnant lady, and the lady will be having the very smooth, I mean to say, delivery of the baby. That is how once upon a time, what was a poison or what was a toxin, and it has become the, I mean to say, medicine after some time. So more than 50 formulations are made from same toxic material produced by the claviceps. I list out these are the various I'm going to say formulations. These are the formulations used in treatment of several disorders. And now I'm going to talk about some of the beautiful kind of forms what are microscopic. In the earlier form of the slide, what I discussed is the mushrooms, which are microscopic forms. And earlier, the mushrooms are having the long history back. The mushrooms were earlier given, I mean to say, food of warriors, I mean, it's a soldiers, people who participate in the war are given the mushrooms for making them more strengthened or more power to increase the power. So this was believed in the Greek civilization as well as in Roman civilization. This was mushroom was considered as a food of gods, and after that, centuries later, the Chinese have, I mean, say people feel that mushroom means it is a Chinese origin, but these mushrooms have the history from Greek, and it has come from Greek to Rome, Rome to China. Then last China considered it as a healthy food. And this is how the picture, how the, let me say, in 1651, the rediscovery of mushroom has occurred in the particularly France, where a melon crop, I mean to say watermelon, we can see now today watermelon or muskmelon. So a melon farmer was seeing that in his heap of manure, organic manure, a typical kind of button type of growth was there, and he was surprised to see that, I mean to say, in France it was not very well known, and it was growing without any kind of interpretation. Suddenly that growth made the farmer to be fantasize, and in his fantasy that farmer took that the outgrowth, and he took that outgrowth and he grown in the typical same kind of substrate, and he found that a plenty of mushy growth was appeared, and this whatever the mushroom growth was there, he sold this to the particularly Parisian restaurants, and it was introduced in the Paris restaurants as a typical dish. So it was by 1707. There were gardens where lot of mushrooms were grown in the French community. So slowly from French community, the other parts of the world also started making the conventional practices to grow this mushroom in the as a crop. So till 1955, a lot have been improved, and by 1975 commercial mushroom cultivation was practiced. So more than 2000 species of mushroom exist in nature, and only 25 species are accepted as food. Among them, some of them I will be showing you the pictures. So these are the mushrooms, and we are also fortunate to have these mushrooms in our grocery stores. You can see the mushrooms of various varieties here given: Pleurotus, Agaricus, Lentinus, and so many Ganoderma, and various forms are also there. I will be specially focusing on this type that is shown here, Ustilago mayadis. So the approximate I'm going to say nutritive value of these mushrooms are listed and listed here, and they have told that the mushrooms are rich source of protein. You can see, you can have the comparison of the protein diet. So the protein in this diet is very high, and carbohydrates are also very high, and surprisingly the content which is not good for health, fat, is very less. You can see over here the number of when you say the reducing sugars content and total sugars, here it is compared, and fatty acids. And especially we know that all teachers we know that the fatty acids are of two varieties, that is saturated fatty acids and unsaturated fatty acids. So when you talk about the saturated fatty acids or saturated fats are not good or safe for the health, but unsaturated fats are very good, or you can say PUFA, polyunsaturated fatty acids. When you talk, they are very good, or they are also called as essential fatty acids, EFA. These fatty acids are very good for the health, or they are good cholesterol or good fats for the body, which protect the body from various kind of antioxidant activities and so on. So you can see that lot of I'm going to say lot content of unsaturated fatty acids is there, which makes mushroom and good kind of diet. And again, it is very important to discuss one more thing, that what happened is in the 1993 there was a sudden high medals, high number of highest medals from China. Then people started wondering, and they were doping tests were performed for the Chinese athletes, and most of the Chinese athletes were found to be negative for the doping test. Means no Chinese athlete was found to take any kind of drug or any stimulant. Without even taking any stimulant, people were surprised at how China or Chinese athletes have got so many medals, and how the China has topped in the Olympics. So then slowly some coach has revealed the truth, and the truth is this fungi or caterpillar fungi, that is called cordyceps or Ohio cordyceps, which is the caterpillar fungus. And this fungus is typically not even mushroom, but still it is just because of its fruiting body, typical fruiting body. You can see that this fungi will infect the insect, and it will kill the insect larva, and then it will form the fruiting body, and this fruiting body is used as the particularly medicine, or you can say it as given as a drug, and it is called as kida jadi in India. So more than 400 species of this are available, and usually this cordyceps is very famous in the Chinese traditional medicine and also Tibetan medicine, and it is normally found to grow in the months of April to August, and then it is parasitic in nature. As I told you just now, it will infect the insect larva called as Hippialis, and after infecting the larva of Hippialis, what it happens is the larva will be dying, and the fungus will be growing out of the entire body of the insect, and then we can see that the typical appearance of this we say caterpillar fungus is visible. So this is how the picture where life cycle of cordyceps is shown over here, how it infects the insect larva and how it becomes into a particularly caterpillar fungus is shown over here. And there is a big list of caterpillar fungus here. We can see the uses of this caterpillar fungus. And now I am going to talk about a typical. So there is a small video. Please watch this video.
Sir, are you playing the video, sir?
You're not seeing able to see the video?
No, sir.
Oh no. What is visible there and again?
Your screen, sir, a delightful fungus. It is titled as Ustalgiomydes infected or corn as a delicac. That's all, sir.
Oh no. How to get this video then again on the screen? I have to share this, huh? Wait. Yeah, I'm sharing the screen. Wait. Yeah, I'm sharing the screen now. Are you able to see it now?
Yes, sir. Yes, sir. Now it's okay, sir.
Okay then. Thank you.
Okay, sir. Thank you. Sir, you have to enable system audio so that we can even listen to that video's audio, sir.
Okay, I'll make it.
Okay, sir.
80% of all insects live in jungles. Few are more successful than the ants. There can be 8 million individuals in a single hectare. But jungle ants don't have it all their own way. These bullet ants are showing some worrying symptoms. Spores from a parasitic fungus called cordyceps have infiltrated their bodies and their minds. Its infected brain directs this ant upwards. Then, utterly disorientated, it grips a stem with its mandibles. Those afflicted that are discovered by the workers are quickly taken away and dumped far away from the colony. It seems extreme, but this is the reason why. Like something out of science fiction, the fruiting body of the cordyceps erupts from the ant's head. It can take 3 weeks to grow, and when finished, the deadly spores will burst from its skin. Then any ant in the vicinity will be in serious risk of death. The fungus is so virulent, it can wipe out whole colonies of ants. And it's not just ants that fall victim to this killer. There are literally thousands of different types of cordyceps fungi, and remarkably, each specializes on just one species. But these attacks do have a positive effect on the jungle's diversity, since parasites like these stop any one group of animal getting the upper hand. The more numerous a species becomes, the more likely it will be attacked by its nemesis, a cordyceps fungus.
Galaxy S21. Il a l'air différent. Il est différent. Tu peux filmer en 8K, encore mieux qu'au cinéma.
So again, you were watching that
We like to think we're in control, that our minds are our own. But that's not.
hope I can start the sharing my screen again.
Yes, sir.
Is it visible now?
Yes, sir, it is visible.
Delightful fungus.
Yes, sir.
Okay, so what was the story? What I was showing you, I think you have got it or not, but again you can go back to the same YouTube link and you can watch the thing. There are some ants called carpenter ants. What is the specialty of this carpenter ants is these ants are growing in the tropical forest. Plenty of these ants are available except in Europe. Other than Europe, all of the continents, even India, we can see these ants are there. So they will be growing on the tall trees. And what is the specialty of these ants is the ants will move from the ground to the top part, canopy part of the plant, and they'll start feeding on the green leaves, and that will make the leaf affected. And these ants are going to make a heavy damage in the tall trees. So these ants are controlled by the infection of the, just now what we told, cordyceps mushroom, a fungi, ascomycet fungi. So these ants are infected in the body, and the ant is expected to die because of the fungal infection, and the whole ant body will become the, I'm going to say, source or substratum for growth of the fungi, that is Ohio cordyceps. Earlier you're seeing was a different fungus that belongs to the same genus cordyceps. So this fungi will be the one which is going to control the population of ants. And this is how we can say that fungus or whatever the fungi is there, these typical fungi is going to control the population of this, I'm going to say, devastating ants or insects. It's a typical kind of, we can say, this kind of mutual benefit from the fungus is a ecosystem service. It is best example of ecosystem service, that nature itself is correcting its, I mean to say, nature itself correcting the problem, or it is solving the problem of the particularly the trees, or we can say the plants, whatever the plants are there, the plants are checked to be healthy by their own protectants. You can see that God has created such a wonderful system. You can now believe that how the particularly tiny insect causing a huge damage to the plant is being controlled by a typical enemy or its typical pathogen, that is the cordyceps. And again, this cordyceps is also very, very, very beneficial for the, I'm going to say, the fruiting bodies of these are picked and used for various kind of treatments in a traditional medicine. So that was what, I don't want to exaggerate the situation. Now I want to go to a typical kind of fungus. That is delightful fungus. Delightful fungus means this is a fungus which is making you to have water in your mouth. Why this making water in your mouth is the thing I'm going to talk to you. This fungus is very well known to have its own name, and it is the, I would say, rich in antioxidants. You can see that a lot of antioxidants are present in this fungus or fungal fruiting body, which is going to infect the corn and corn cobs. You can say these are the corn cobs, and this is the smut disease. It is a smut disease caused on the corns, and this corn cobs having infected with the Ustilago species will give you a fruiting bodies, and these fruiting bodies are again consumed. You can see the bread, rolled bread, you can see, and you can see even the particularly grocery shop tin which is sold. And this is what is the name of this in the market. So this is it is written in a typical way, and it is pronounced in a typical way. It is pronounced, though it is written with the H, but it is pronounced as V Tala Kocha. This is the name of the typical fruiting body in the market, Vitala Kocha. So Vitala Kocha is a typical grocery item in the grocery stores of Latin American people and the Mexican people. These people would like to have the soup of this fruiting body, that is Vitala Kocha. So the Vitala Kocha is again shown its dietary, I mean to say nutritive value. Here it is shown, and this is the to say fresh harvested corn with the fruiting bodies. And this is how the ingredients are mixed. Different ingredients are basil, sweet basil leaves are there, and again some different culinary items are there. They are mixed, and then you can see that how beautiful soup. I don't know whether we don't have the taste, but in Mexican and Latin American, this soup is very costly, and again it is very good for the health. And this is how the culinary dish made from the fruiting body of the infected corn infected fungi. So it is all what the, I would say, deadly part of fungi, and again also it is the delightful part of the fungi, and also the help, I would say, how the fungus is going to be the healer, life-saving one. All these activities are shown, and I want to ask the organizers, what is actually my time to get finished my talk? I think by 3 o'clock I have to wind up, isn't it?
No,
Or
sir, you
3:15?
can continue, sir. 10 more minutes, you can continue, sir.
10 more minutes. Yes. Okay. Thank you. So I will talk for 10 more minutes because I have to come to my part. What part is I'm going to talk, what we are doing actually. All these things I'll stop, and I will tell you the plants are actually inhabited with a lot of microorganisms in them, and one such beautiful example of colonization of microbes, colonization of fungi. When you talk about the colonization of fungi, we can see there is a term called endophytes. What are the term endophyte means? Endophyte means nothing but the fungi are going to inhabit inside the particularly plant host without causing any kind of typical symptomology. So the term endophyte was actually proposed by a long back person called Anton von D. Berry, and this is a recent paper what I have seen that directly they have made the fluorescent micrograph or fluorescent pictures of the endophytic fungi colonizing the various plant forms, that is bryophytes, pteridophytes, gymnosperms and angiosperms. So equally, not only the fungi are going to colonize here, even the bacteria are found or shown. Bacteria are also shown to colonize the same plant systems, where we can see the fluorescent or green. What a green part is there. The green part is the visualization. The tissues are being colonized by the fungi. Here, bacteria also shown equally in the particularly way by using the different fluorescent dyes. Here are the names of the fluorescent dyes what were used to stain the sections of the various plants to visualize the partners, fungal partners or bacterial partners in the particularly plant systems. And what are these endophytic fungi? Again, this name D. Berry, and before D. Berry, there was again person called Henrich. Henrich was in 1809 and D. Berry in 1866. And recently, one person who is well known in this field is Orlando Petrini. He made the endophytes, very, limelight, brought to the limelight. So, what is this? This is a story how this endophytes in recent times have been rediscovered. Though endophytes have been told long back in 1800 itself, but recently what has happened is more important. There was a problem in cattle. What was the problem in the cattle? The cattle were having a toxin problem, toxicosis problem. And why this cattle were having a problem? The cattle were having a problem because of eating some grass. What was the grass? This is the grass what the cattle were made to eat. The grass was this grass was fed to the animals, cattle, grass-eating animals, and they found that these animals were having some typical disease. And till that time, the diseases were known. Diseases were well known to have the phenomenon. They should follow any disease caused by infectious agent should follow Koch postulates. But this disease was not at all fitting in the Koch postulates. And later on, they have seen slowly that the grass was having a typical kind of toxic item. And this toxin was not due to the plant or grass itself, but there was some fungi present inside this grass, host plant, and it was causing and it is causing to release some typical toxin, and this toxin was causing the grass to become poisonous or toxic to the animals. So that is how people come to know that there is presence of endophytic fungi, and these endophytic fungi will help the host plants to escape or to develop the defense mechanism. They have developed the defense mechanism like this. So like this, it was the first time people have come to know again about the endophytic fungi and their metabolites, which are going to protect the plant system. So this is the how this grass was it's famous, and its fungi was also very famous. So these fungi later on were started after the big episode of this toxicosis. People started studying the fungi for various these endophytes for various other purposes. So in such case, they have found that both beneficial kind of fungi will the benefit these fungi are beneficial not only in defense but also in the various other activities. The activities are given over here, and now you can see how the fungi has the role in both the endophytes have the role in both the positive and negative. So all these are there, and basically I want to focus on mainly this part, and this part is more important. What I have done is yesterday I think our moderator has introduced me and told that I have done one UGC project. So in that UGC project, what was my target actually? In that UGC project, my target was all this what I have shown to you. Not many works I have carried out, but I did diversity. I did the biodiversity of Apocynaceae family. So what I might have done, is here you can see that I have collected the plant material, selected plant material of Apocynaceae. Then I have gone for the isolation of fungi, and these fungi were purified and they were identified based on morphology. And we have performed some of the growth promoting activities in some of the plants. So this is how the endophytic fungi are being studied for various various activities like cytotoxic activity, antioxidant activity, antimicrobial activity, antiviral activity, anti-diabetic activity, immunosuppressive activity, and so on. There are the various protocols. We have to follow various protocols for characterizing the typical kind of tests we have to perform, where we are going to screen the genes also genetically, because morphological characterization is not sufficient. Now we have to go for molecular characterization. So molecular characterization will have a big story how we will perform all these things. So with this, I want to finish my presentation. So if somebody is really interested to ask questions, please you can ask questions. Somebody genetics or somebody has raised the hand once upon a time. If that person want to ask any question, please you can ask your question. My presentation is done now. I have finished as per my time. My time was to finish till 3:30. I have to continue, I think. Okay.
Sir, if you want
Somebody
to
want to
continue,
ask questions for 15 minutes, I can ask. I can tell you the question. Its answers on this endophytes or any any concept about this fungi. Then it will be very. I will be fortunate to get some questions from somebody.
sir, if you want to continue, sir, or else we'll go for questionnaire.
Yeah, it is very important that we should get some kind of some questions. It will be better.
Okay, sir. I have unmuted all the participants. If you want, you can unmute yourself and ask the questions.
Sir, Good afternoon, sir. I'm Sunaina from Suryapet. Sir, I have a question. When you're working on these endophytic fungi, how do you grow them in the lab? Do you really cultivate them on the plants on which they are endophytic, or can they be grown on different medias in the lab?
You are a teacher or you are a student?
I'm a teacher, sir.
Then fine. I think you are very familiar with the how fungi are cultivated in the lab, right? Initially, in this story, what we do is we will first take the healthy plant part, either it may be a
Yeah.
leaf or it may be a stem or whatever part we are targeting. Sometimes we
Okay.
go for leaf, sometimes we go for stem, sometimes we go for root also. So
Hmm.
first we take the healthy leaf material, and then
Hmm.
by taking the healthy leaf material, bring it to the lab and we'll then sterilize it. You know that how we sterilize the things in tissue culture. Similarly,
Right.
we'll make the surface sterilization, and after
Okay.
surface sterilization, we'll make the short fragments of this plant part, and then we'll be preparing the media in the plate that is protected exposed agar. Usually, we'll use protected exposed agar, and on the protected exposed agar, the sterile fragments, the whatever sterile fragments are there, these sterile fragments are overlaid on the surface of the protected exposed agar plate, and then we'll be keeping aseptically in the BOD incubator at a temperature between 25 to 27, and then we will be monitoring the growth in the plate. So usually what we do is they can be a slow grower and they can be a fast grower. If it is a fast grower, within 48 hours we can see lot of growth will come, and we can see that growth is appearing. So as soon as we see that the fragment growth surrounding the fragment, then what we do is we'll uh, uh, take the sterile needle aseptically, we'll transfer it into the fresh plate, and a new plate we'll inoculate it, and we'll allow the sometimes we'll do 3-point culture, sometimes we'll do single point culture, and then we'll be making the fungi to grow in the isolated plate, and from there we'll be taking that plate for the further characterization of the fungi. Thank you for asking question. Thank you so much. Thank you from my whole heart. Actually, I was
Sir,
expecting.
actually, my question is this endophytic fungi, whatever the toxins or alkaloids they are producing, are they producing in association with the plants when they are in endophytic association?
Yeah.
Or it's the inherent property, even when they grow it in the plates, they can produce such
Please wait,
compounds?
please wait. I'll tell you. So there is an interesting concept. Uh, generally the gene transfer. I think yesterday we heard about the gene transfer, right? Yesterday we heard about a gene transfer. There is a vertical gene transfer and horizontal gene transfer. When the fungi go, these fungi are not very uncommon. These are common fungi. They are found also outside the plant. So when the fungi enter the plant host system, there is a possibility. There is a, I mean to say, there happens a gene transfer from the plant host to the fungi. So this whole transfer of gene is from plant to fungi or fungus is characterized. Fungus is called horizontal gene transfer. Just because of this horizontal gene transfer, the fungus is able to express its system, and produce the toxin. Hope you understood. Or
Yes, sir, I got the answer.
shall I elaborate?
Yes, sir. Thank you.
Usually the same thing happens in case of agrobacterium. What we do in the agrobacterium is we are going to make a vector, and so much exercise we'll do. But in nature also there is a gene transfer occurring. What we do in agrobacterium is artificial gene transfer, but what it is happening here is a natural transfer of gene between two systems, that is a plant and the fungi. Because of that only the fungi is able to produce such kind of typical compounds produced by the plant. Thank you. Any such questions are asked again by somebody else?
Good afternoon, sir.
Yes.
Can we differentiate poisonous mushrooms and edible mushrooms only by their physiological appearance, sir?
Based on because they are morphologically it is I'm to say there are some mushrooms which are actually characterized for edibility, like 25 out of 2000, 25 are generally characterized. So only out of 25, 4 or 5 are sold in the grocery shop. So as you are asking the difference between the poisonous mushroom and the safe mushroom, identifying based on their appearance, yes, sometimes based on the, I would say, typical pigmentation, as you are telling about the morphological identification, so typical pigmentation itself is alarming us to characterize whether it is a toxic form or non-toxic form. Their pigmentation are the important source of characterization at this point today.
Okay, sir. Thank you.
Thank you so much. Anybody else? Just 25 more minutes are there. I think as per my time, I have to go down till 3:45.
If any
If somebody
further
else, some
questions are
students,
there,
some
they
students
are,
yes, yes.
dear participants, if you have queries, please post it. Dear participants, please ask your queries if you have.
From your college also, some student is interested to ask anything like any something.
Okay, sir, just a moment, I'll let you know. Sir, as I could not get any of our students ready to ask now, so can I sum up the session, sir?
No, wait a minute, because we have to talk something about the academies, and I will finish off the thing. After that, we can end up the things.
And that's all, sir. I just want to glimpse your lecture, sir. That's all. So thank you, sir, for justifying your title, what you have chosen as fungi as friends or fees. You have clearly explained that fungi can be friends as well as fees if they are used in various vaccine developments and medicine developments from years together, and they are even used as fees as they can cause immigration, discovery, ergotism, holy fire, like that. You have posed a very good and live examples. Even you have shown a beautiful video which can show us the development of mushrooms and the dyed ants. So thank you for explaining it in a clear way, sir. Thank you so much.
Sneha, madam. Sneha, madam.
Yes, sir.
Uh,
So we
Sir,
are,
good evening, sir.
yeah, good evening.
So,
So I will give
yeah,
you
Vasami, ma'am, can you
one,
take the principal's
yes, yes,
aspect?
yes.
one small input about the academies, because it is very important for us to tell people whosoever are attending here. We should tell what are the academies and what are they doing actually, because it is partiality from our side if we don't praise. See, equally we should praise the society for making this kind of institutions and empowering the women in employment as well as the education sector. So we equally thank the society for this kind initiative by government of Telangana, and equally we for this today's meeting we should feel very fortunate, and it is our duty to let me say remember once what are these academies, and the young generation especially should know what is academies actually. So I will little bit talk for 5 more minutes, and then I'll let end the things, because my time was given up till 3:45. So I will be summing up before 3:45. So I am going to talk first of all about the three academies. The first academy of our support is the National Academy of India, National Academy of Sciences India, that is called NASI. The National Academy of Sciences India is called NASI, and this was the most earliest academy which was found in the year 1930. So initially this academy, that is NASI, National Academy of Sciences India, was called Academy of Sciences of United Provinces Agra and Oudh. And at that time, Meghnad Saha, yesterday I was talking about Meghnad Saha, astrophysicist. So at that time, he was the chairman of this academy. I mean, academies are not after the independence, but academies are existing before the independence. So many people were there in this list. What were the academy was there, the initial academy, where Meghnad Saha was the first president, and in the year 1990-91 they have celebrated the Diamond Jubilee session, and lot of scientists, great scientists, were the fellows of this academy, and presently more than 1856 fellows are there, among which we had two fellows, we witnessed two fellows, Dr. Surendra Gaskadbi and Dr. Rita Mulharkar, were among the fellows of this particular academy. And this academy has got some of the visions, like promote scientific and technological research for societal welfare, and they will publish the proceedings, journals and transactions, and they will organize the meetings. Like we had a meeting on scientific and technological problems, and then they have the constituted committees and bodies which will focus or undertake the scientific works for public importance. And then this academy is cooperating with various research organizations in India as well as abroad. They are organizing a science library also with them, and president of this National Academy of Sciences India is Professor G. Padmanabham. He is the former director of Indian Institute of Science, Bengaluru. This was a brief about the present one of the academy, and now comes about the second academy. The second academy is the most important academy where we are asking the funding for such programs. So it is the Indian Academy of Sciences, Bengaluru. So what is so special of Indian Academy of Sciences, Bengaluru is, after 1930, in the year 1934, Sir Chandrasekhar Venkat Raman, Nobel laureate of time, has established or founded this academy in Bengaluru. And in the year 1934, 27th of April, he founded this academy, and he was the president of that academy at that time. And presently this academy is more, I mean to say, more, I mean to say, doing activities like academy fellowship, annual and mid-year meetings, science education programs, sponsorship of discussion meetings, the Women in Science panel, repository of scientific publications of academic fellows, oral I mean to say, oral history archives, then committee of scientific values, Raman Chair Professorship, Jubilee Chair Professorship, publications of academy, and then contributions to science, contributions to science policy. So here we can see that whatever our program is there, science education program, in the science education program, there are some of the important events like the lecture workshops, refresher courses, and also summer fellowships. I request all the students as well as their teachers to please check out this in Google for summer fellowships supported by academies. So there is an application open for the particularly summer fellowships. What is summer fellowship actually? Summer fellowship is a program supported by the 3 academies. You have to apply it online, and you have to select one of the mentor from the list given there. You can go to the website and check the list of the mentors, and after checking with the mentors, based on your interest, the mentor will have its own area. Somebody is working on Arabidopsis, somebody is working on something like wise. When they are working for some kind of, I mean to say, research area, they have to pick up the research area, and they have to communicate with the particular person. And then if he is willing, then we have to fill his name in the mentorship. And then what does academy do? The academy do is they will support, I mean to say, the stipend. They will pay stipend based, I don't remember what is the present stipend. So they will pay the stipend, and they will also help. They'll also help for staying over there. The stipend covers the particularly incidentals as well as the particularly stay in that particular area. I mean, it's accommodation and all they will support. So for the final year graduate students, it is a good opportunity to track this, and they can try to go for some kind of summer fellowships and get the thing. This is the most important event that students should avail. The student should avail this facility, and try to get the good research experience along with your academics. So the main focus of this workshop is to make awareness among you to know what academy is actually doing for the benefit or the pursuit of science. So that among 3 programs, the student summer fellowship program is very important, and even teachers can also avail this summer fellowship, and teachers can also apply. Teachers have some quota and students will have some quota. So it is a duty of teacher to get motivate and make the girls also, make the students also to motivate for this program. So please don't forget to check out for this summer fellowship program, what is supported from Bangalore. So you need to apply it online. No need to go anywhere, and no need to say write anything to them. So directly you can have this program on the particularly online thing, and you can go offline and work with the eminent scientist of the given list. That is more important to be known by you. And the last academy what I wanted to share is the Indian National Academy of Science, that is INSA. INSA was actually started in the year 1935, I suppose. And by 1935 it has started, and it has also have the objective, various objectives, like earlier academies have the objective promote the scientific knowledge in the country, and coordinate with other societies and institutions. Like today we have coordination with them, and interest of I mean to say, and scientists, they will promote the promotion and the safeguard of the scientists, and they will also, I mean to say, constitute national committees for to perform the public and government activities, and they will publish the journals again. And like this, this academy, the Indian National Academy, is located in the New Delhi. So like this, with this briefing, I think that still 10 more minutes are there for your valedictory. So I want to express my deep sense of gratitude towards the academy, the office, who was very, I mean to say, who are very kind enough to support us in this program. Though the budget was very low, but still the institution today has reached one step ahead, and peak to conduct online virtual meeting. So if this meeting was offline, it might be too colorful, I hope. And we want to express our deep sense of gratitude towards chairman of the science education program, Professor Murthy sir, and the coordinator of this program, science education program, CS Ravi Kumar sir. We express also a deep sense of thanks to Ravi Kumar sir, who has been kind enough to support us and make this event so colorful. And we equally thankful to the personal assistant of Ravi Kumar sir, Geeta ma'am. Geeta ma'am is one who looks after all these activities, or she will be updating online all these programs. Our name, our institution name, and name of our today's coordinator, Sneha madam, is also there in the lecture workshop website in the sponsored programs. So we can have that benefit also. So maybe the money what we have got is less, but we have got a good encouragement and good startup for the institution, new budding institution. We could do such a good activity with a huge number of participants. I could see yesterday a big population, but I don't know what happened today. We are seeing very sick population. Whatever it may be, the population is more or less. We could able to perform such a big, wonderful activity. With these few words, I want to sum up my conversation. Thank you, and over to the organizers. Thank you so much.
Thank you, sir. Thank you so much for your description here. Many students might have got the information regarding that, but still, even I would like to give a brief report about that. I really take this privilege of sharing about these academies.
So you're having your valedictory, you are having your valedictory. Till that time, you can take the feedbacks. You can
Yes,
collect.
yes, sure, sir. Yes, sure, sure, sir.
Your valedictory, you plan in your my
It is
part
there,
was,
sir.
my part was to present my talk and present the supporters who supported us, right? So whatever feedback is there, you can use that for your valedictory, and you can ask your students and teachers to give the feedback. And meanwhile, it will be 4, and then you can start your feedback valedictory program, and you can run the show like that.
Yes, sir. Sure, sir. Sure, sir. Sure,
Madam, I don't know this madam's name, but Sneha madam's name only I know, and some botany faculty people I know. Thank you so much, all of you. I will be able to take permission to quit the Zoom meeting. Thank you so much once again for all of you.
Thank you, sir. Thank you so much, sir.
And I am very happy to announce that we have got 2-week refresher course sanctioned today itself. A while before, we have come to know from the academy office that we are going to have 2-week refresher course for life science teachers, which is supported by the academy. So I hope that our we will write to the society office so that online virtual refresher course could be useful for the academy. Society. Sorry, again I'm repeating academies. So in my happiness, the thing is, we expect that many teachers will be listening to me, and also we will write to the society office also that they can there are only 30 participants can join our course. It is a refresher course. I think you are newly joined and you might be knowing the value of refresher course and how this is an online refresher course. Now UGC has cut down the duration of refresher course from 3 weeks to 2 weeks. So this 2 weeks refresher course is also having equal, what UGC, your HRDC center is going to make the program, similar program we are going to organize. And we feel that many more teachers could be again be in our 2 weeks program. So we will again get in touch with society people, and we'll see that many more teachers can get benefit of this refresher course in knowledge as well as the certificate.
Thank you, sir. Thank you so much for your concern, sir. Thank you so much. Congratulations also, sir, for this.
Thank you so much. So I'll take a leave now. Thank you. Bye.
Thank you, sir. Thank you. Thank you, sir. There's a small announcement to the participants. There will be a link shared down in the chat box or in the WhatsApp group. Please give the feedback, and then you will be receiving the certificates within 2 days. Kindly fill the feedback. If necessary, even if you feel like you can even express your views about this workshop. Now, before beginning the valedictory, I request our participants to express their valuable thoughts on this workshop. Kindly, dear participants, please give any feedback regarding this workshop.
Hello, ma'am. I'm a lecturer in genetics from Mahendra's College. The program today for the 2 days is fully engaging. We are very happy that we are given an opportunity to meet such eminent scientists on this platform. So the workshop was mostly on genes and genomics. So I was very much interested in all of it. I posed multiple questions and I got wonderful answers for it. I'm sure that the course of genetics in our colleges will be expanded and more students will be interested because they now understand that the future of medicine is all based on pharmacogenomics and genetics. So definitely this workshop, I'm hoping that many students will be inspired to take up this course, maybe if not now for coming academic sessions. Thank you very much, ma'am, for giving us this opportunity. Congratulations to everybody. I mean, this is a wonderful workshop. I think your mic is muted, ma'am. I can't hear. Thank you, ma'am. Thank you.
Thank you, ma'am. Thank you for your valuable thoughts. Any other participant, please? We would be delighted to receive any feedback.
Good afternoon, madam.
Good afternoon, ma'am.
And this is Badrikali from Barangal West Degree College, madam.
Good afternoon, ma'am. You're welcome.
So the program is so useful not only for the staff but also to the students. We hope this type of program should be conducted in all over the degree colleges. And thank you so much for being a participant of this program. Thank you, ma'am.
Thank you, ma'am. Any other participant, please? Now, actually, these academies have sponsored many such lectures throughout, but this is for the first time our college, Telangana Social Welfare Residential Degree College, Nizamabad, got an opportunity to organize these academies virtual lecture workshops. So we really feel, happy, fortunate, so that's why we would thank academies in trusting and giving us such a platform and exploring the knowledge like this. Thank you so much. And any other, please? Any other feedback? And any thought, if you feel like giving any suggestion, we are open to suggestions, please. Shall we wait for some more time for the feedback? I think somebody is raising hand. Somebody with the name Sirisha, Janavi, but could not speak. Ma'am, please give your feedback.
A delightful good afternoon to madam and sir over present there. And this is in front of you. This is Pandana from Residential College of Degree from Nizamabad. This is from BJC second year. First of all, I felt very happy and a very pleasure to meeting you and attending this online workshop. Thanks for inspiring me and motivating me to enter into this research field. I really felt very happy and I gained so much knowledge how to enter into research field, and it helpful for me to my further studies. And thanks everyone, madams, for present over there to inspiring me so much to enter into the research field. Thank you, madam.
Thank you. Thank you for your feedback. And any other, please? Even yesterday, we got many calls from our colleagues and from other side saying that this workshop was very informative, and so they were very much delighted in participating in this. Thank you so much for the feedback, and thank you so much for your support and encouragement.
Hello, ma'am.
Yes, ma'am.
Yeah, the workshop is really very helpful, ma'am. So really we enjoyed all the lectures. Very eminent personalities were given the lectures, and really we thoroughly enjoyed all the sessions. Actually, whatever the recent technologies that are there, recent developments in the biotechnology and crop improvement regarding cancer medicine, really all the lectures were very fruitful, and you have given us a great opportunity to listen to the eminent personalities. The lecture was very fruitful, ma'am. Thank you.
Thank you, ma'am. Thank you so much for your feedback. It gives us immense pleasure to take this feedback. Other participants, kindly, it will be a great motivation for us. Your feedback would be a great motivation for us. So I would like to brief up about this Academy's lecture workshop. These academies, these are sponsored workshops, like sponsored by 3 academies: Indian Academy of Sciences, Bangalore; Indian National Science Academy, New Delhi; the National Academy of Sciences, Allahabad, India, has sponsored many such workshops. But this is the first workshop that we have organized here, and we are really fortunate for that. So Indian Academy of Sciences, Bangalore was first founded by Sir C.V. Raman in 1943. Its aim to promote the progress and uphold the cause of science in pure and applied branches. The Academy holds major activities that include publications of scientific journals, special volumes, recognition of scientific talent, and perks up the science education. So we would look forward to many such opportunities, and we would be delighted to host such kind of workshops. And any other feedback, please? I think some other. See, kindly check once an announcement to the audience again here that there is a link shared in the chat box. Kindly fill the feedback that is there in the link, and you will get the certificate within 2 days. Some of our colleague has written a very good feedback here. Very informative and interesting session, ma'am. Even though I'm not related to this course, the lecture on Hydra and cancer treatment are more catchy and found interesting. Thank you, ma'am. There's another feedback from Ms. Sandhya. Good afternoon, all. This is Sandhya Rani. It is really a valuable workshop. Thank you. Thank you, ma'am.
Good afternoon, ma'am.
Good afternoon, ma'am.
This workshop is very informative, ma'am, and thought-provoking. We were very much delighted in listening to this eminent scientist, which inspired us to continue our productive research work in useful areas.
Thank you, ma'am.
Thank you, ma'am. Thank you. Great
Thank you
session.
so much, ma'am. Thank you so much, ma'am. There's another feedback from Ms. Farhana Begum from GG College saying that thank you for conducting such webinar. Thank you, ma'am. Dear participants, to get your certificate, please give us feedback through the link. You'll get the certificate. There's one more feedback from Miss Vindhya. Interesting session, madam. Please send the YouTube link for lectures if possible. Yes, sir, sure. Yes, ma'am. Definitely, we'll share you the YouTube links for this lectures. Definitely. Thank you, ma'am. Thank you for the feedback. Another feedback from Asima Fatima. Congratulations for conducting insightful and informative workshop. In future, we expect hands-on workshops. Sure, ma'am. Definitely, ma'am. Thank you, Vindhya, ma'am. There's another response from Miss Jessica. Good afternoon, ma'am. Workshop is very informative and interesting, and it will be very useful for me. Thank you for conducting such an awesome workshop. Thank you so much, Jessica, ma'am. You can give any suggestions, ma'am, if needed. If you feel like we need to change something or could have been much more better if we implement some more else, you can give some suggestions also to us. Sumalata, Miss Sumalata has also given a good feedback here. Good afternoon, all. The workshop is so informative. Thank you for conducting this workshop. Thank you, ma'am. Thank you, Sumalata, ma'am. There's another feedback from E. Lahri, Miss E. Lahri. Thank you very much for organizing such a wonderful and informative workshop. We are looking for many more such great opportunities which enlighten the young generations. Thank you, Miss Lahri. Thank you. Asima ma'am has congratulated Sushma ma'am and Sneha ma'am for giving the gist. Thank you, ma'am. There's another feedback from Usha, Miss Usha. Good evening, everyone. Thank you for conducting such a wonderful workshop. Lecturers were so knowledge loaded. Great teamwork. Thank you, ma'am. Thank you for the feedbacks in the chat box. There's another feedback from Miss Supriya. Good afternoon, all. The webinar was so excellent. Thank you for conducting such an informative workshop. Thank you so much, Miss Supriya. Yes, there's someone who has raised their hand. Yes, Ushasri ma'am can speak here or can post a message in the chat box. This is another message from Miss P. Pavani. Great teamwork, madam, and very informative session. Thank you, Pavani ma'am. And there's another message from Swati madam, Swati Tulasi madam. Good afternoon, ma'am. It's really an amazing informative workshop. I have gained the collective information. Thank you, ma'am. Thank you so much, Swati madam.
Really, the workshop is very informative and very useful to all the students. Even non-science students also come to know about mushroom, whatever culture in Hydra. Really, those sessions very interesting. And I have much interest about this workshop. It is worthwhile. Thank you, ma'am.
Ma'am, thank you so much. There's another feedback from B. Swapna. It is very wonderful workshop. Thanks for giving such an opportunity to us. Thank you so much, ma'am. There's another feedback. Congratulations to all the faculty for organizing such a wonderful workshop which deals on modern trends in biology. Great speakers delivered excellent lectures. Thank you all, ma'am. Thank you, Ramya, ma'am. There's another feedback from Nirmal College. Good afternoon, ma'am. Thank you for giving this opportunity to listen to eminent person lectures. It is very useful to all, ma'am. Thank you so much, ma'am. There's another feedback from K. Rajita. Good afternoon, ma'am. It's great workshop. Thank you for conducting such a workshop. Thank you so much, Ms. Rajita, ma'am. We are looking forward for any suggestions here. Kindly please post some suggestions. Nirmala, ma'am, thank you so much for your feedback. There's another feedback from Mr. Ramana N. Good evening to all. It's an amazing informative workshop. Thank you so much, sir. So kindly stay tuned. We have valedictory beginning shortly. There's someone saying that link is not opening. Please kindly check that. Is it for Badrakali, madam? Is it for the feedback or the YouTube link? Feedback link. Okay, feedback link. Badrakali ma'am is unable to access that feedback link. Ma'am, we'll get back to you, ma'am. Kindly check after some time. Yeah, some other is also saying that it is showing there's a mistake. Consult host after sending the feedback. Okay, ma'am, we'll definitely. Please check after some time. Thank you, Anita ma'am. We are actually waiting for the dignitaries to arrive. So once they join, we'll immediately start with the valedictory.
Sneha, I have joined through Shiba Roy.
Okay, sir. We'll check once.
Laptop.
Okay, sir. Okay.
Right.
Thank you, sir. Thank you.
I have few internet issues, so I may not my.
Sir, your voice is not clear, sir.
Yeah, I have few internet connectivity issues where I am now.
Okay,
So I enable my video, but my audio is enabled.
Okay, sir. Okay, sir. Okay. Sure. Thank you, sir.
Hello,
Yes, sir. Om
this is
Prakash.
Professor.
Yes, sir.
This is Professor Om Prakash.
Yes, sir. Yes, sir. Thank you, sir. Welcome, sir. Welcome to the session, sir.
Right. Thank you.
So now the end of the story is the new beginning for many others. On this note, let us enter into the valedictory. But before entering into that, I would request our principal, Dr. P. Snehalata, to few words.
వినండ్రా నేను వదిలేస్తా అనుకున్నావా ఏంది
Welcome to the valedictory ceremony. Now let's begin the valedictory. May I now request our zonal coordinator, Professor Om Prakash sir, to address the attendees. May
Thank
I
you.
request? Thank you, sir.
Thank you. Dr. Snehalata, the principal of TSWREIS Nizamabad. Dr. Praveen Mamidala, Joint Secretary, Higher Education, TSWREIS and Hyderabad, and the team of learned colleagues from Nizamabad College, and learned colleagues from other colleges, and teacher participants of this workshop. At the very outset, I congratulate the principal of the college and our team of teachers in taking the initiative to organize 2 days virtual lecture workshop on the theme of trends in modern biology. I appreciate the efforts made by the principal and our staff. In addition to that, we must be thankful to the 3 academies, the prestigious academies of sciences. One is Indian Academy of Sciences, Bangalore. The second one is Indian Sciences Academy, New Delhi. And the third one, all these 3 academies having sponsored this program, that speaks the importance of this program to improve the teaching and research skills of the young faculty of the residential degree colleges. Of course, I am not a man of sciences, but nevertheless, as a teacher of a university, I know the importance of biological sciences. And particularly, the importance of biological sciences have grown and received wider importance in the context of latest pandemic, Corona, COVID-19. Definitely, as scientists of biological sciences, you play a greater role in tackling such diseases which come as part of the changes in our lifestyle. And I have been listening to the feedback of various teachers before I joining this session. So it is very clear that the teacher participants have got an excellent opportunity of listening to eminent and experienced scientists from different walks of the life. And definitely this goes very well, and this will contribute to the improvement in the teaching and research skills of our faculty. But As a university teacher, I would like to say something here. Just participation in virtual workshop, lecture series workshop will not result in or will not yield good results unless we put whatever we have learned through this workshop into practice. And I believe that the lectures delivered by eminent speakers will motivate the younger generation to improve the knowledge of their domain subjects. And not only that, their lectures would motivate them to take up more and more research projects, and in turn, they will enlighten the teaching learning environment in the residential degree colleges. So I request all the participants, pay attention to whatever you have learned in these 2 days virtual lecture series workshop on the trends in modern biology. So I thank and I express my gratitude to the organizers of this workshop for having invited me to speak a few words as part of this valedictory session. And I wish the TSWREIS in Islamabad would take up more and more such workshops, not only in biological sciences, but other sciences. Because, Snehalata is principal, and this is second online workshop. This is second workshop I am made part of it. And when she was the principal at Kottagudem, so she organized a seminar in the microbiology. But apart from microbiology and biological sciences, there are other sciences. So definitely, I look forward to see many more such workshops in other fields of sciences, maybe physical sciences, chemistry, and even social sciences also. I wish them all the best, and thank you, all the participants. Thank you very much.
Thank you, sir. Thank you so much for the valuable talk. We would definitely take your suggestion and we'll try to implement that shortly, sir, again. Thank you so much, sir. Thank you for your gracious presence. Now I request Ms. K. Anita, lecturer in chemistry, to speak a few words.
Good afternoon, one and all. Firstly, I thank our principal ma'am for giving me this opportunity. The workshop is very informative and thought-provoking. We were very much delighted in listening to the eminent scientist, which inspired us to continue our productive research work in useful areas. We look forward to many such talks in the future too. Congratulations to the team. Thank you.
Good afternoon to everyone. This is E. Sandhya Pridarshini, Department of Botany, TSWRDC Nizamabad. It's an immense pleasure to share my experience on this lecture workshop. It is a very prestigious program as there are eminent and great scientists involved in this workshop, giving their experience on their research on various fields in modern biology with very good explanation, which is very easy to understand by the students. This is an auspicious occasion to know the recent modern trends in biology, as it includes inspiring lectures on their extensive research work in developing genetically modified crops, regeneration capacity of hydra, and it is very interesting to listen again how the long DNA thread is bounded in the small cell. Human Genome Project, how can a genetic disorder can be treated through gene therapy, and Phytoremediation, how it can help in absorption of heavy metals and radioactive elements from the soil, and eco-restoration by using vetiver grass by demonstrating live projects. Thanks for giving me this opportunity. Thank you.
Now, I would like to take the privilege of inviting our source of motivation, our Joint Secretary, Sir, Dr. Praveen Mamidala, Sir, to share a thought. You're welcome, sir.
Yeah, thank you, ma'am. Thank you very much. I hope to you. Am I audible?
Yes, yes, sir. Your voice is breaking. Actually, it's still we can. Yeah, we'll listen to it, sir.
Okay. Okay, friends, let me congratulate the entire Nizamabad Telangana Social Welfare Residential Degree College team for their
Thank you, sir.
superb efforts, and also I congratulate the principals Neha, the general coordinator, Professor Om Prakash, and all the teachers who are behind this project. I am sure that you have made an excellent job by conducting such virtual sessions. First of all, let me thank the convener and the coordinator, the professors who are behind this project, particularly the INSA, the National Academy of Sciences, and the Indian Academy of Sciences. First of all, let me thank Corona, because of Corona, we met within no time. If at all it is not Corona, that we cannot do such a wonderful program within no time, because I have experience of organizing such academy lecture workshops before. It is a very Herculean task, inviting professors and bringing them to one platform. But because of this online virtual system, now it has become very easy. At the same time, I thank the resource persons, Dr. Surendra Gaskadbi, Dr. Susan, Dr. Rita, and Dr. Ahmed Abdul Halim Khan, all these professors who have enlightened our students, because I know as a biology student, evolution is a confusing word. Evolution is a Greek word to many of the science students. So they fear a lot about evolution. But whenever you go through our yesterday lectures and today lectures on evolutionary dynamics, I think all of the students have enjoyed a lot. And thanks to Dr. Susan for her journey from Mendelian's laws to the genomics project till date, and also Dr. Rita highlighting on the next generation sequencing technologies. Because of some reasons in India, still we are lagging behind, but the abroad, US and Europe, they are doing great in terms of the NGS tools. They know how best to use the NGS tools in terms of transcriptomics, proteomics, and genomics work. So I am sure that even the Abdul Halim Khan explaining about green revolution to gene revolution, what are the various milestones till date is really awesome. I thank on behalf of Telangana Social Welfare Residential Educational Institution Society. Today, actually, Secretary Sir Dr. R.S. Praveen Kumar, IPS COP, who has transformed the entire society to always question, which we forgot. I would like to convey an important project to the resource persons today, to Dr. Surendra ji and Dr. Susan ji and Dr. Rita ji, that we have initiated a project called as the Telangana Social Welfare Research Council. The Research Council is trying to fund or trying to support our teachers to come up with projects, with an outstanding projects, out-of-box thinking projects. If at all they can propose a good project, we are going to fund them 10 lakhs. Because now DST, DBT, AICT and other funding agencies, they're not able to fund money because of some reasons. But to fill that gap, we convinced the Telangana government, and the Telangana government has sanctioned 6 crores of rupees to our society to research project in terms of minor research project. Another best thing to add this is for students also. If the student of a degree second year comes up with a very good research question, we are going to fund them 50,000 rupees. I think even in the USA also, we do not see such projects, but thanks to our visionary leader, our secretary, Dr. R.S. Praveen Kumar sir, who has given us freedom to do such projects. So, ma'am, and Dr. Surendra ji, and Rita ji, and Susan ji, all you have to help us in mentoring our teachers, in mentoring our students in future, how to ask a question and what is the rationale and how to resolve that question. Your help is badly needed for our students. These days, we don't find the greatest committed and dedicated scientists like you people. And on behalf of society, again, once again, I thank all the resource persons and I thank the organizers, the entire team of Nizamabad Residential Degree College, and also the students who participated in this program and all teachers who participate in this program. Thank you very much. Thanks a lot. Over to Sneha.
Thank you, sir. Thank you so much for your motivation. Thank you so much for your inspiration and your deep insight. We consider your thought as a mission and would definitely strive hard to fulfill that. Thank you so much for your support, sir. Thank you. Last but not least, I request our Vice Principal, Ms. K. Lavanya, to propose a vote of thanks.
Good evening, everyone. As the saying goes, all good things come to an end, leaving us with memories and knowledge imbibed. We now come to an end of the 2 days Science Academy's virtual lecture workshop on trends in modern biology. On behalf of TSWRDC Nizamabad, I take this opportunity to propose vote of thanks to those who are directly and indirectly contributed to this workshop organized by our institute. First, I would like to thank Science Academies for accepting our proposal and given these wonderful opportunities. The Academy's contribution to science and society is immeasurable. Our heartfelt thanks to convener of workshop, Dr. Surendra Gadkarbi, for gracing his important work and willingness to share his valuable time. We really enjoyed your sessions, sir. A special mention to Dr. Rita Mulhekar, ma'am, for taking time from her busy schedule and made our young minds to know recent technologies of gene therapy and personalized medicine for cancer. We are grateful to Dr. Susan Ipan, ma'am, for sharing her knowledge on crop improving approaches. From bottom of my heart, we would like to thank Dr. Abdul Halim Khan, sir, who has been the backbone of this workshop. Because of your efforts, sir, we could get a chance to listen to lectures from such eminent personalities. Thank you once again, sir, for guiding us and helping us throughout the workshop. Thanks to Limbadri, sir, and Professor Nasima, ma'am, for adding beauty to the workshop. A big thanks to our Secretary, sir, Dr. R.S. Praveen Kumar, sir, always been inspiration for us through his actions. Thank you, sir, for being guiding force. We would like to extend our thanks to Joint Secretary, sir, Dr. Praveen Mamidalam, for his support and guidance. Special thanks to our Zonal Coordinator, sir, Professor Om Prakash, for helping out the Institute to fare better. I'd like to thank our Principal, ma'am, Dr. P. Snehalata, who has been the guiding post for the conduction of the workshop. Right from planning, enrollment, and execution of the workshop. She has been proactive and coordinated with all the teams. Thank you, ma'am, for your dynamic leadership. We would have succeeded in this workshop because of your dynamic leadership. A special thanks to all committee members who were strongly determined for the huge success of this workshop. Our thanks goes to technical and non-teaching staff, teaching staff for their unflinching support and coordination. Special thanks to technical staff from head office.
Our heartfelt thanks to all the participants for their active participation throughout the session for the past 2 days. I'm sure this workshop has delivered what it intended to. Thanks again to everyone for making this workshop a huge success. Thank you all participants for being with us for these 2 days. Thank you one and all for your support and coordination. A small announcement, we have shared the feedback link with the participants. Please fill the feedback. You will receive the certificates within 2 days. Thank you one and all. Bye bye for the session.
Thank you, everyone. Thank you, Zonal Coordinator, sir. Thank you, Joint Secretary, sir, Pavit Mamidala, sir, for joining us. Now, let's put this session to an end. And I request everyone to raise for the national anthem.
जनगण अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उत्कल जलधि तरंगा तब शुभ नामे जागे तब शुभ आशीष मांगे गाहे तब जय गाथा जनगण मंगलदायक जय हे भारत भाग्य विधाता जय हे जय हे जय हे जय 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 हे.
Thank you.